Greetings from University of Veterinary and Animal Sciences. I am Dr. Sadia Nawaz, working as Assistant Professor in Institute of Biochemistry and Biotechnology. Today I am going to present a part of my PhD work. Title of my presentation is Wnt10A mutation associated with a complete Odonto-Onycho-Dermal Dysplasia syndrome. Odonto-Onycho-Dermal Dysplasia is a skin disorder and it comprises a heterogeneous group of developmental abnormalities which constitute about 200 clinical entities. It's a rare syndrome and it's characterized by severe hypodontia, nail dystrophy, smooth tongue, dry skin, keratoderma, and hypohidrosis of palms and soles. The family that we had a surgeon from a Punjab region. We had spent 5 generations in this family, and the clinical presentation of this family showed that the affected members had one of the affected member had sole of right foot in individual 7, and at the age of 37, which was indicating, which was presented with the palmoplantar, with the plantar keratoderma. There were dystrophic toenails and smooth tongue with reduced papilla. It was there, and the panoramic radiograph of the affected nail member at the age of 39 showed that the most of the permanent teeth had third molar excluded, and the upper jaw there are 2 permanent central incisors and 2 permanent premolar with conical crown shape. In the lower jaw, there were 4 permanent premolars with reduced crown form, the bottom arrows. And the scheme of studies, when we have to study the the inherited disorders, we go for the linkage analysis, which includes, first of all, pedigree analysis, where we can see the what is the mode of inheritance or the pattern of inheritance in this family. Then we look for the candidate genes which are linked with that region, and we do the exclusion mapping. And if we don't see any linkage, then we go for the genome-wide screening and the statistical linkage analysis later again turn to the novel mutations, novel loci, and at times novel genes. Nowadays, NGS has made this much easier than going through all these steps. The family that we had is certain, we spanned 5 generations in this family, and there were 6 individuals which were affected by Odonto-Onycho-Dermal Dysplasia, and the inheritance pattern was autosomal recessive. The degree of consanguinity was very high in this family. When we designed primers for the candidate genes, which were tightly linked with the skin disorder, we had found that our gene, our region that was linked, that was associated, tightly linked with this disease was gene Wnt10A. Sequencing of the Wnt10A gene showed the position that was changed or the transition that we had observed in the chromatogram that was in the nucleotide replacement C to T, which had ended up in A to V change or replacement. This homozygous Wnt10 missense mutation that we had seen in all 4 affected cases of the OODD syndrome. The first report of missense mutation, it is that is showing a mutation in the Wnt10A gene, and it has resulted into alanine to valine substitution. Uh, Earlier, a Wnt10A nonsense mutation at position 697 G to T was reported in 6 distantly related OODD patients. When we looked at the phylogenetic analysis, the situation was highly conserved across species. Uh, to summarize our results, we had observed this with Win10A and W10A mutations in OODD. The homozygous missense mutation is associated with a complete OODD phenotype, contributing to the classification of ectodermal dysplasia. And the findings emphasize the importance of Win10A in the formation of ectodermal appendages such as hair, teeth, skin, and nails in the humans. And the identification of human phenotypes with the other Win10A mutation or possibly mutation in the downstream members of this signaling pathway will be important to further understand the developmental process of the ectoderm. These studies will be improving our current knowledge about Win10A in ectoderm development for both diagnostic and biological importance. The work may prove helpful to study gene function and learning more about the pathophysiology of OODD disease, and in long term, this can lead to a better understanding of the genetics behind acute dysplasia that will contribute to improved diagnostics, risk assessment, therapeutic strategies, and to develop improved treatment models for these disorders. The results of this study will also help to provide genetic counseling to the carrier families for precautions of affected birth, and the study may lead to a marked reduction in the number of severely affected children born with inherited disorders of skin in the Pakistan population. This is the map of consanguinity, and thank you for listening.